So again, hi everyone, my name is Maria Kardakova and I'm a PhD researcher at the University of Surrey. And today I'll be presenting our research on gut microbiome and major depressive disorder and how we conducted the investigation on the microbiome composition between cases and control in our study. We also investigated how is there any genetic predisposition contribute to depression risk. So this study is a collaborative effort with my supervisors, Aije Dermirkan, doctor, and Professor Inga Prokopenko, and the Atlas Biomed study group. And and the gut microbiome, as you may know, it's shaped by multiple factors, including diet, medication use, genetics, and environmental exposure. And we know that the gut microbiome plays the vital role in not just the immune system function, but also neurotransmitter production and metabolism. So, however, its relation to major depressive disorder is still being unexplored, MDD, or major depressive disorder, is a widespread and deliberating mental health condition that affecting approximately 5% of the global population, with high of prevalence in females. And it is characterized by persistent sadness and cognitive impairment and even loss of interest to daily activities. So it can also affect the lifestyle of the individual. So recent studies suggest that the gut dysbiosis is also linked to MDD, but findings are often inconsistent, and some studies indicate reduction of microbial diversity in MDD, while others do not. So the role of specific bacteria taxa also varies, and it's making it crucial to use larger scale and well-powered studies to clarify those relationships. So this is one of our aims with the current study, is to identify microbial taxa that associated with MDD, assess microbiome diversity differences, and explore causal relationships between gut microbiome and MDD using Mendelian randomization. So we also evaluate the predictive potential of microbiome risk score for MDD by integrating microbial signatures and lifestyle, clinical, and epidemiological data. So let's discuss our data set that we worked with. It contained the gut microbiome data, lifestyle data, and dietary insights from over 7,000 individuals from Eastern and Western Europe that who filled the over 200 item questionnaire, shared the dietary insights, and uncovered some of the medical history. So we also received their 16S rRNA gene sequences. And so this data set allowed us to examine the interactions between gut microbiome composition, lifestyle, and MDD risk across cases and controls. So in overall, we had 822 individuals with diagnosed depressive disorder and over 6,000 healthy controls. We also splitted our set into two cohorts, discovery and replication, in order to conduct differential abundance analysis. So we utilized MASLIN2 and ANCOM-BC2 models with further performed meta-analysis. So we also did the adjustments for age, gender, geographical region, and batch effect. So we utilized one of the latest tools that proposed with the with a group of scientists who actually investigate the large-scale data sets. So here you can see the results of our meta-analysis, where we utilized cases and controls running with a Maslin-2 test. And you can see the 16 bacteria genus taxa that positively or negatively associated with their depressive disorder. And further, we utilized the UNCOMB-C statistical analysis in order to investigate the differential abundance among cases and controls, and we see 9 taxa that associate with the MDD. And we also did the crossover between 2 analysis and 2 meta-analysis of 2 different methods, and receiving in total 9 taxa of crossover. I'm sorry if you cannot see properly the names of this taxa, but we will upload the slides so you can see them in more details maybe after that. But these 9 taxa contain Rhombusia, Clostridiums, Sensu stricta group, Agatobacteroids, Pictonophilus, and Coccrococcus, victivalis, which have a negative association with